All right, let's jump right into this incredibly fascinating breakdown of Parkinson's disease. In this explainer, we're going to unpack the science behind this condition, tracing it from the physical symptoms we can actually observe all the way down to the microscopic and even genetic levels. It's a true macro to micro masterclass, and honestly, it's going to completely change how you view this neurodegenerative disorder.
To really wrap our heads around this complex condition, we're taking a structured zoom-in journey today. We'll start with decoding Parkinson's, move to the basal ganglia circuit, look at the pathological hallmark, explore the gut-brain axis, and finally, crack the genetic blueprint.
So, Section 1, Decoding Parkinson's Disease. We're starting right at the surface level, looking at how this disease actually manifests in a patient.
Historically, Parkinson's has always been defined by what we can physically see. You're probably already familiar with those classic cardinal motor features, things like bradykinesia, which is basically just a general slowing down of movement, along with that recognizable resting tremor, rigidity, and postural instability. But here's a really surprising fact. The non-motor symptoms are equally critical. We're talking about a sudden loss of smell known as anosmia, or maybe acting out your dreams during REM sleep, or even severe autonomic and gastrointestinal issues like constipation. And, as you'll see a bit later in this explainer, these aren't just random side effects. They are massive, crucial clues to how this disease actually begins.
Okay, moving to Section 2, the basal ganglia circuit. Now that we've seen the outward symptoms, let's zoom directly into the brain to look at the exact circuit board that's malfunctioning.
Movement in the human body is controlled by this incredible biological circuit board. In a healthy brain, signals flow right from the cerebral cortex into the basal ganglia, specifically into the striatum, which is made up of the putamen and caudate. Think of it kind of like a relay race. The signals get modulated by deeper structures, like the substantia nigra pars compacta, and then they feed right back through the thalamus to tell your muscles exactly what to do. It's a continuous, beautifully orchestrated loop.
But this right here perfectly illustrates the core mechanical failure. Normally, dopamine from the substantia nigra acts as a stabilizer for two distinct pathways, balancing the movements you want to make with the suppression of the ones you don't. But in the Parkinson's state, that dopamine is heavily depleted, and without it acting as a vital, stabilizing current, the indirect pathway literally goes into overdrive. This dopamine loss leads directly to hyperactivity in both the subthalamic nucleus, or STN, and the internal globus pallidus, the GPI, that basically locks down or inhibits the thalamus. And the end result is that classic poverty of movement we just talked about.
Which brings us to Section 3, the pathological hallmark. If the circuit is broken, let's zoom in a million times closer to the cellular level to see exactly why those dopamine wires are frying.
What's absolutely fascinating here is that we don't just guess this happens. We can physically see the localized destruction. Take a look at the left side of the slide. That's a cross-section of a completely healthy midbrain. Notice those dark pigmented streaks of neuromelanin? That pigment is found in healthy dopamine-producing neurons. Now, look at the Parkinson's brain on the right. You can physically see the devastating loss of those dark neurons. The literal factory that makes the dopamine has just been wiped out.
And the wild thing is, we don't have to wait for an autopsy to see this anymore. We can actually image this functional loss in living patients using PET scans. On the left is a normal healthy scan. You see it glowing bright yellow and orange in the striatum, where those dopamine transporters are highly active. But on the right, in the Parkinson's patient, just notice the dramatic reduction of that vibrant dopamine signal, especially in the posterior putamen. The circuit is quite literally running on empty.
So, what's actually killing off these cells? Well, zooming into the surviving neurons, we find the true villain of our story. See that pinkish circular mass inside the cell? That is an intracellular eosinophilic Lewy body. It's a toxic, clumpy inclusion packed with misfolded proteins, primarily one called phosphorylated alpha-synuclein. And as these Lewy bodies start to multiply, they slowly and systematically suffocate those delicate dopamine neurons from the inside out.
Now for Section 4, the gut-brain axis. We know what the Lewy bodies are, but where do they actually come from? Let's explore a really fascinating theory about how this pathology spreads. This right here is one of the most cutting-edge concepts in neurology today, the break staging hypothesis. Basically, the break hypothesis suggests that this misfolded alpha-synuclein acts almost like a prion. A prion being an abnormally folded protein that forces healthy proteins it touches to misfold as well, like a domino effect.
The theory proposes that this misfolding doesn't actually start in the brain at all. Instead, it begins way down in the peripheral autonomic nervous system, or even the gut, and then physically travels upward into the brain via a prion-like chain reaction. This timeline perfectly maps that path of destruction, and it connects right back to those non-motor symptoms we covered earlier. The pathology likely starts decades before anyone gets a clinical diagnosis. 20 years prior, those misfolded proteins might begin in the enteric plexuses of the gut or the olfactory bulb, which perfectly explains early symptoms like severe constipation or anosmia, that loss of smell. By about 10 years prior, it travels up the vagus nerve to the dorsal motor nucleus, triggering things like REM sleep behavior disorder. And it's not until year 0 that the pathology finally hits the substantia nigra pars compacta, triggering those classic motor tremors we all recognize. It's a remarkably slow, sequential invasion. Finally, Section 5, the genetic blueprint. For our last zoom in, let's look at the very foundation of human biology, the genetic mutations that make someone susceptible to this whole cascade. Now, while the vast majority of Parkinson's cases are considered sporadic, meaning they just happen without a clear family history, get this: up to 15% of cases are actually linked to specific genetic mutations. And studying that 15%, it gives us incredible clues about the fundamental mechanics of the disease for absolutely everybody.
This table neatly organizes the main genetic culprits. We've got dominant genes like SNCA, where mutations directly cause an overproduction and misfolding of that toxic alpha-synuclein protein. Then there's LRRK2, which ramps up kinase activity and is actually the most common genetic form of PD. On the flip side, we have recessive genes like PRKN or Parkin and PINK1, which are heavily linked to early onset or juvenile Parkinson's. These recessive mutations essentially break down mitophagy, which is the cell's ability to clear out damaged mitochondria. It's literally like the cell loses its ability to take out the trash.
But perhaps the absolute most critical genetic discovery recently is the GBA1 mutation, which happens to be the strongest known genetic risk factor. It perfectly ties everything we've talked about together. GBA1 mutations severely impair lysosomal glucocerebrositis activity. In plain English, they cripple the lysosome, the cell's internal garbage disposal. Because the disposal is broken, all that toxic alpha-synuclein piles up. But here's the kicker. That piled-up alpha-synuclein then further blocks the lysosome from working. So it creates this devastating vicious cycle where the trash just keeps building up, destroying the cell's ability to ever clean itself.
Which leaves us with this final, really inspiring frontier. If we now fully understand the macrocircuitry, the misfolding protein, the prion-like spread from the gut, and the underlying genetic pathways, could tomorrow's cure look entirely different than today's? If we could somehow target the gut microbiome or clear that alpha-synuclein protein a full decade before it ever reaches the substantia nigra, well, we might be closer than ever to therapies that stop this disease before the very first tremor even begins. It's a huge thought to chew on as you digest this masterclass. Thanks so much for joining me for this explainer, and keep learning.